All right, hello and welcome everyone. My name is Riley Frost, and I'm a biochemistry major at the University of New England. And I'm going to be sharing my capstone research project with you all. That was a requirement of um, University Organic Chemistry for the fall 2020 semester. Unfortunately, I was not available to participate in the lab portion of this project. But I am going to try my best to get across the results of our research project, what happened and why it happened. As you're seeing right here, this is a overview of our entire poster. And the main question that we were trying to answer was, how does a low temperature affect the isomeric product of the alkylation of sodium saccharin? So a little background and significance. Sodium saccharin is the crystallized form of saccharin, which is an artificial sweetener used in beverages and food, and it was first produced in 1879. The main difference between sodium saccharin and saccharin is that saccharin has an NH bonding group, and sodium saccharin does not. Instead of an NH bonding group, it just has a nitrogen, and then it has a somewhat floating sodium ion. Some of the physical properties of sodium saccharin are that it is stable at a pH of 2 to 7 and at a temperature of 150 degrees Celsius. The molecular weight is 206.18 grams per mole. The melting point is 224 degrees Celsius. And some hazards are that it is flammable and can irritate the skin. So previously, we did a class experiment that identified how a temperature range of 70 degrees Celsius to 90 degrees Celsius can affect which isomer is produced. And this temperature range only resulted in the N-ethyl product being formed. It has been identified that at lower temperatures, the rate of an SN2 reaction can increase with a decrease in temperature, and that stereoisomers can be selected based on reaction temperature. In order to identify which temperature range change would, in order to identify which temperature range changes which product gets formed, we decided to cool the reaction to around -3 degrees Celsius to 0 degrees Celsius in that region. This was done using a brine ice water bath, which is similar to that of a water bath in setup. Low temperatures also result in molecules being less energetic, meaning that their electrons are more localized to an atom with the highest electronegativity. For our experiment, this is significant because it means that electrons will favor the oxygen atom more than the nitrogen atom, which will result in a higher electron density. This is beneficial because then the nucleophilic reaction will occur at the oxygen atom instead of the nitrogen atom, and this will result in the O-ethyl product being formed. As we know what conditions produce N-ethyl saccharin, our rationale for this experiment was to attempt to produce O-ethyl saccharin using new conditions. Our main research question for this experiment was, how will dramatically lowering the temperature of the nucleophilic reaction using an ice bath affect which saccharin isomer is produced? Will the reaction temperature affect the product yield? And our hypothesis to answer this question was, if the nucleophilic reaction temperature is lowered, then the O-ethyl saccharin isomer will be produced. As you can see here, these two figures are our reaction and proposed mechanism. Figure 1 shows the overall reaction for the substitution reaction of sodium saccharin salt, which will hopefully yield the O-alkylated product at 0 degrees Celsius. And figure 2 shows our proposed mechanism for the substitution reaction of sodium saccharin salt, which will also hopefully yield the O-alkylated product at 0 degrees Celsius. The mechanism is a bimolecular nucleophilic substitution, also known as an SN2 reaction. The electrophile is ethyl iodide, the nucleophile is sodium saccharin, and the leaving group will be iodide. So some of our experiment methods. 0.92 grams of sodium saccharin and 2.5 milliliters of dimethylformamide, also known as DMF, were added to a 25 milliliter round bottom flask with a stir bar. The reaction flask was lowered into a brine ice bath at a temperature of around -3 to 0 degrees Celsius, and it was left to chill for 15 minutes. A thin layer chromatography sample was taken right when this was lowered into the water. After the reagents were chilled, 0.4 milliliters of iodoethane was added. A TLC sample was immediately taken, and it was sampled every 10 minutes until the reaction ran for 20 minutes. After spotting the TLC plate, the reaction was deemed completed and allowed to run for an additional 10 minutes. 30 milliliters of ice water was added to the reaction flask after these additional 10 minutes to help our product solidify. However, the precipitate did not form, and liquid-liquid extraction with ethyl acetate was performed. The ethyl acetate layer was dried with magnesium sulfate. The organic layer was purified with the rotary evaporator and was set aside to crystallize. And also to facilitate the drying process, a vacuum pump was also used. The white crystals were analyzed via infrared spectroscopy, and percent yield was calculated. And below we have a couple of figures, which are pictures from our lab. Figure 4 is one of my group mates, Matt, inspecting the rotary evaporator during concentration. Figure 5 is the rotary evaporator setup. Figure 6 is the vacuum apparatus that was used to help dry the precipitate. And figure 7 is the experimental setup in the brine ice water bath. Some extra instrumentation details. The rotary evaporator that we used was a Bucci rotary evaporator R100 with chemically resistant vacuum pump. The infrared spectrometer we used was the Nicolet IS5 IR spectrometer. We used sodium chloride salt plates, and the samples were prepared as a cast film, which is a thin film of compound that's coated on the salt plate. And a couple of things about our thin layer chromatography analysis. We used aluminum back plates. The plates were coated with a silica gel stationary phase doped with a fluorescent indicator. Visualization was made possible by shortwave UV light at 254 nanometers, and this was followed by immersion in a iodine sand chamber. And the eluent we used was a 1-to-1 hexanes ethyl acetate. And again, below is figure 5, which shows our rotary evaporator setup. So this is our results. As you can see, there are 3 IR spectrums, and then there's a table that shows the percent yields for each trial and an average. So to get these IR spectrums, we dissolved the crystals in a flask of DCM, and then we pipetted this onto the salt plates and let that evaporate. And we put the plate with the which we had let the DCM evaporate from into the IR spectrometer, and it was able to get a reading. And so these are the 3 readings that were produced. Figure 9 is one of the IR spectrums from cohort B. It is a little bit different than those from cohort A, which are shown in figure 10 and figure 11. But this is most likely due to contamination, which I will talk about in a future slide. But in figure 11, we have cohort A's infrared spectrum. And then in figure 10, we also have another spectrum from cohort A. And then for our percent yields, the reactant mass was used to calculate the theoretical yield, and the product mass was used to calculate the actual yield. And then we divided the actual yield by the theoretical yield to get percent yield. And our average percent yield was rather large And this was most likely due to cohort B using too much ethyl acetate and an error of measuring the flask with and without the solvent. So between cohorts A and B, a total of 5 experiments were performed. Originally, we were looking at 6 replicates, but due to my circumstances, we were not able to do that. So only 5 were completed. But in continuation, infrared spectroscopy was used to determine the identity of the product of the alkylation of sodium saccharin, and percent yield was calculated. Based on the infrared spectra, the product is most likely saccharin, which is shown in figure 12, rather than O-ethyl saccharin, which does not support the original hypothesis that O-ethyl saccharin would be produced at dramatically low temperatures. From the peaks of the IR spectra, which are shown in figures 9, 10, and 11, we derived the following functional groups. At around 3,400 centimeters to the negative 1, we had an NH stretch, which would be part of an amine. At around 2,900 centimeters to the negative first, we saw an sp3ch stretch. At around 1,700 centimeters to the negative first, we saw an sp2 carbon oxygen double bond stretch, which would be part of a carboxylic acid. And then around 1,300 centimeters to the negative first, we observed an sp2 carbon nitrogen stretch, which would be part of an amine. Since these functional groups that I just listed off are not found in O-ethyl saccharin, this is what led us to believe that saccharin was produced, because these functional groups are found in saccharin. And also the product was insoluble in water, and that is one of the properties of NH saccharin. However, based on data from another experiment, saccharin appears as a white crystal, but the product that we obtained was not a white crystal. It was a more oily, kind of half solid type of product, and it had a little bit of yellowish color to it. This led us to believe that contamination or error may have been present in our experiment. And as I said earlier, one replicate in cohort B, shown in figure 9, during liquid extraction, may have been contaminated by water and ethyl acetate, and that's why the infrared spectrum is slightly different than in cohort A's results. The contamination during liquid extraction may have also led to improper formation of the product after rotary evaporation. But based on our findings and reevaluation of our original hypothesis and mechanism, we think that lowering the temperature of the reaction may have led to a completely different reaction mechanism instead of the original SN2 reaction, and that may be the reason why we did not get the O-ethyl saccharin product that we hypothesized we would. In conclusion, dramatically lowering the temperature of the reaction did not produce O-ethyl saccharin. It appears that saccharin in its NH form, which is shown in figure 12, was the product that resulted. A future investigation would use further data collection to confirm this result. Other possible methods of analysis would include NMR, melting point analysis, and GCMS. However, these methods weren't available to us during our experiment because of the oily nature of the product or technical difficulties with the instrumentation. We attempted to try melting point analysis, but our product was just not solid enough to fit in the capillary, and because of this, we could not do melting point analysis. For further research in melting point analysis, one would expect a melting point range around 228 to 229 degrees Celsius based off other literature. This could then be used to determine if our product was pure saccharin or something different. In the future, this research could be brought in a direction to further investigate the mechanism of the reaction that occurred in this experiment. In-depth analysis of the product could provide insight into how and why saccharin was produced at this temperature. We are unable to comment on the mechanism indefinitely due to our lack of expertise on the subject at this time. However, we believe that an examination into the possibility of a different elementary step causing these results may be helpful. More trials definitely performed at the same temperature, as well as others performed at a wider range of temperatures with complete data collection and analysis would definitely provide insight into this new investigation. Below is a list of references. And for acknowledgements, we would like to thank our professor, Amy DeVoe, for coaching us through all of our research, teaching assistants Hunter, Chloe, and Ben, and the University of New England School of Mathematics and Physical Sciences for allowing us to use the facilities and materials that they provided us with in support of our research. Thank you. That is all I have for you today. If you have any questions, feel free to reach out.